Okay, welcome everyone to our chapter on the lymphatic system. Remember that this works as an adjunct to the immune system, and so the two systems are involved in pathogen defense. The lymphatic system refers primarily to the movement of um, tissue fluid that exists between cells through a series of vessels and eventually into a series of lymphatic organs that will scan that fluid for the presence of pathogens or cancer cells and then generate a physical and chemical attack designed to remove those cells from the body, render them useless before they get a chance to produce disease. The immune cells are the cells that assist with that process by producing the chemical and physical attack. So we're going to describe what lymph is and where it comes from. We'll describe the pathway of lymph in its return to the blood, and we'll describe how lymph is moved through the lymphatic vessels, and we'll talk about the different lymphatic organs, the spleen, the thymus, um, and the lymph nodes. We'll talk about the components of nonspecific resistance against disease. We'll discuss the immune response, and we'll explain the mechanism of cell-mediated immunity and antibody-mediated immunity. We'll compare primary and secondary immune responses and talk about what immunizations are and what they accomplish. We'll talk about how the immune system is involved with the rejection of transplanted organs and tissues. And then finally, we'll touch on some lymphatic and immune disorders. So, what does the lymphatic system do? Well, it filters fluid that's between cells, scanning it for the presence of pathogens. This fluid makes up about 15% of the total body fluid, which is in the lymphatic system at any one time. It also maintains the water balance in the body, and it's involved in the absorption of fat from the digestive tract through special structures of the lymphatic system. They're called lacteals, and it works with the white blood cells to protect us against disease. So you can see here sort of a diagrammatic of how lymph is collected and how it flows back into the circulatory system. Basically, the tissue fluid between the cells, the interstitial fluid, works its way into lymphatic capillaries as a result of um, pressure differentials between the lymphatic capillaries and the tissue fluid. The fluid is then propelled through lymph vessels that promote one-way flow through the presence of one-way valves that are basically spaced throughout the system. Eventually, this tissue fluid reaches a lymphatic organ. Generally, it's a lymph node, and within the lymph nodes are macrophages and lymphocytes who will scan the interstitial fluid that enters for the presence of cancer cells or pathogens. The macrophage, of course, will eat the pathogens and then present antigens derived from those pathogens on the cell membrane surface, and that will serve as a trigger to the lymphocytes to generate now an army of cells that will generate a physical and chemical attack designed to wipe out the pathogen before it produces disease. The filtered fluid is returned to the circulatory system near the right and left subclavian veins, and as a result rejoins now the body's plasma. So this is important because if this flow into the lymphatic system is impeded, that tissue fluid will build up between the cells, and that'll produce a condition known as lymphedema, which can impede circulation and compromise immune function. Lymph is the fluid that flows inside the vessels. It's derived from blood plasma. Capillary leakage causes blood proteins to enter the interstitial fluid, and an increase in interstitial fluid osmotic pressure causes excess fluid to accumulate in the interstitial space. So remember again that osmosis is simply the movement of water down its concentration gradient. The lymphatic vessels pick up and return the accumulated fluid to the bloodstream after it's been filtered, and as a result now we are disease free most of the time. This process prevents lymphedema in body tissues. So here you can see that lymph is a one-way flow from the capillaries to the interstitial fluid, and then from the interstitial fluid into the lymphatic capillaries, which then empty into lymphatic vessels, which empty into lymphatic ducts, which eventually empty into lymphatic trunks, which empty into the circulatory system once the lymph has been filtered. So it's an ever-increasing diameter in each of those different vessels. The smallest are the lymphatic capillaries. These are blind-ended tubes with valves that allow fluid to enter, provided there's a higher pressure in the interstitial fluid than in the lymphatic fluid that's in the vessels. This layer of filtration slits is the result of overlapping endothelial cells that are stabilized by protein attachments that open into the vessel when there's a pressure differential. So there's also within these vessels valves that only allow movement into the capillary and along the capillary towards the lymph nodes, all derived from endothelial tissue. So they don't open in the opposite direction. So in this sense, it's similar mechanically to the valves in the heart that promote one-way flow of blood through the organ. Same thing is true here. Lymphatic vessels, or lymphatic capillaries, merge to form lymphatic vessels, which are similar to veins in structure. You can see valves in lymphatic vessels promoting one-way flow. The lymphatic vessels merge to form lymphatic trunks that drain large body regions. The lymphatic trunks drain into lymphatic collecting ducts that empty directly into the circulatory system. The right lymphatic duct drains the right side of the head and neck, the right arm, shoulder, and thorax, while the thoracic duct drains the remainder of the body. The collecting ducts attach to the right and left subclavian veins, and this completes the circuit for the fluid. And so the fluid needs to be allowed constantly to flow into the lymphatic system and rejoin the circulatory system. If it doesn't, then the fluid will collect between the tissues and cause lymphedema. So here you can see what the system looks like. The little green lines are the lymphatic vessels and capillaries. The big green lines are the trunks and the ducts, okay, that empty directly into the circulatory system. Here you can see the right lymphatic duct and the thoracic duct. And then you can see the lymph nodes primarily concentrated in regions of the body that drain areas that represent ports of entry for pathogens. So this is why you see lots of these in the axillary region, in the cervical region, and along the respiratory and digestive tract, and in the inguinal region, as well as the popliteal and the cubital region. Okay, so quite a few of these, again, all stationed to drain areas of the body where potentially pathogens could enter. Lymph movement is dependent on outside forces because there's no such thing as a lymphatic heart. We have to move the fluid through the lymphatic system through a combination of skeletal muscle action on the lymphatic vessels and also respiratory movements. Okay, so this is very important. Otherwise, the lymph would pool. So this is actually one of the things that's accomplished in the morning when you yawn, is that you're squeezing lymph through the lymphatic system and promoting, again, filtration of pathogens and cancer cells from that fluid. So the valves prevent backflow of lymph, similar to the way they do in the venous return to the heart. Skeletal muscle contraction compresses the vessels, and that forces the fluid through these one-way valves on its way to the lymph nodes, and eventually it'll exit the lymph nodes and be carried again by lymphatic vessels to the lymphatic trunks, then lymphatic ducts, and then rejoin the circulatory system. Respiratory movements also play a critical role. When you breathe in and breathe out, that presses on the thoracic duct, and that allows fluid, lymphatic fluid, to move through the valves as a way of sort of milking the system. It produces pressure differentials the same way that the skeletal muscle movement does. The lymph is going to move from areas of high to low pressure, similar to the way that we accomplish venous return to the heart, especially from areas below the heart. So these one-way valves and these respiratory and skeletal movements are critical. All lymphatic organs are sites of lymphocyte production. So lymphocytes are made immunocompetent in these regions. They mature here. They may remain in the lymphatic organs, or they may migrate to other sites. If they remain in the organ, they're called what are called fixed lymphocytes, and if they migrate, they're wandering. The lymph nodes occur in groups along the lymph vessels. They're widely distributed throughout the body. They're bean-shaped structures, about 2 millimeters long, and they have more afferent than efferent vessels. So you can see here in the diagram of the lymph nodes, the lymphocyte production areas are these little nodules. So we're constantly producing B and T lymphocytes in these regions, and there are also macrophages in here as well. They always have to work together in order to be effective. The afferent lymphatic vessels empty into lymph sinuses, and then once we percolate through these open areas inside the organ, we rejoin the efferent lymphatic vessels on our way eventually back to the circulatory system. So once we've passed through these nodes, we've removed an extensive amount of pathogenic material and oncogenic material. These efferent lymphatic vessels emerge at the hillow. Remember that that's an indentation in the organ that it serves both as an entranceway for blood into the organ and also an exit for the efferent lymphatic vessels. And there are also nerves, of course, that serve these organs as well. So what do lymph nodes do? Well, they filter and clean the lymph. Trapped substances are then destroyed by lymphocytes and macrophages acting as a team. The lymphocytes can remove cancer cells and pathogens, and the macrophages remove cellular debris, dead or immobile pathogens such as viruses and bacteria. But again, they work together, right? Remember that the macrophages will phagocytize these pathogens and then present them to nearby lymphocytes in order to generate an army of cells designed to physically and chemically attack that particular pathogen, i.e. a pathogen that contains that antigen. So they don't work well without each other. Any nucleated cell in the body also has this ability to engulf pathogens from the outside world. This can promote infection of the cell, but at the same time, the infected cell can present bits of the antigens from that pathogen on the membrane surface as a warning again to the lymphocytes to destroy the infected cell. So essentially they all work in concert to alarm the lymphocytes that there's pathogens in the body. The tonsils are lymphatic tissue underneath mucous membranes of the nasal and oral cavities that intercept and destroy pathogens that enter through the nose and mouth, major ports of entry for pathogens. There's 3 groups of tonsils: the palatine tonsils, the pharyngeal tonsils, and the lingual tonsils. A tonsillectomy is where we remove tonsils that have become so infected with pathogens that the tissue becomes necrotic and then serves as a source rather than a prevention of disease. The thymus is a lymphatic organ that lies under your sternum and in front of your heart. It's rather large in infants, and it involutes and atrophies as we age, but its major function is to serve as T cell school. This is where T cells learn to fight pathogens. The thymus produces thymosins and thymopoietins that promote this differentiation and renders them immunocompetent. And once these T cells graduate from the thymus and move into the other lymphatic organs and tissues, the thymus has now served its purpose and can begin the process of atrophy. And this usually happens again before the end of childhood. The spleen is another lymphatic organ. It's located lateral to the pancreas. Its major function is the differentiation of T lymphocytes. It produces thymosin, which promotes T cell differentiation and division, and also renders the T cells immunocompetent. It's the largest lymphoid organ in the body. It's about 7 centimeters wide and 16 centimeters long, located in the upper left quadrant of the abdominal cavity, again lateral to the pancreas and just below the diaphragm. It serves as a center for lymphocyte production, and it has a large venous sinus that allows blood to percolate in. In the fetus, it's a site of red blood cell formation, so erythrocytes are generated here. But in the adult, it cleans and filters lymph. It serves as a source of lymphocytes and macrophages, and as a reserve supply of red cells. It's also a major site of red blood cell recycling and removal. So this is one of the ways, again, that we recover portions of the erythrocyte so that we can resynthesize them primarily in the red bone marrow. It's also a major site of lymphocyte production. When it is removed, we call this process a splenectomy. Sometimes this happens as a result of trauma to the organ. Sometimes a portion of the spleen is removed either to diagnose it via biopsy, or in some cases to remove tissue that's become cancerous. So here you can see a look at the spleen. There's the organ. The blood again moves in through the splenic artery and percolates throughout the organ through these sinuses. The white pulp is primarily again where the lymphocytes and macrophages are located, while the red pulp is where you'll find most of the erythrocytes. You can see that it's covered by a connective tissue capsule, which again isolates the organ from the surrounding tissue, okay, which is important in order for it to function adequately. Okay, what we want to do now is address the mechanism whereby we are rendered disease resistant. Most of the time, as you know, you're not ill, okay, unless you've got some extraordinarily pathogenic condition. You spend most of your life in a state of health, which is remarkable considering the fact that you live in an environment that is filled with pathogens which are around you, on you, and even in you almost all the time. We're protected against pathogens and foreign substances by a combination of nonspecific and specific defenses that provide a chemical and physical barrier to pathogens getting into the deeper tissues of the body and causing disease. Our nonspecific defenses use the same strategy regardless of the identity of the pathogen. So they're not directed against any particular virus or bacteria. They use the same approach regardless of the identity of the pathogen. These barriers include mechanical barriers like your skin and mucous membranes and their secretions, the chemical action of cells such as granulocytes, phagocytosis, which is the purview of macrophages and dendritic cells, just to name a few, the inflammatory process, and fever. All of these are designed, again, to cut down on pathogens getting into the deeper tissues. So let's talk about mechanical barriers first. Your skin is covered on its superficial surface by densely packed epidermal cells that have tight junctions between them. So they're very well sealed together, and they have an acid mantle that tends to inhibit bacterial growth. So it serves both as a mechanical and a chemical barrier from this standpoint. In addition to that, we have to realize that your skin is constantly being shed and regenerated, particularly in this case the epithelial layers. So this cuts down on the potential for infection as a result of constantly shedding these pathogens off into the environment. Mucous membranes trap foreign substances, and eventually those foreign substances will work their way to the digestive system, where digestive enzymes and hydrochloric acid will render them inert or non-pathogenic. Your tears and saliva and your urine flow act as a way to clear or flush pathogens from these regions, and they also contain immunoglobulins, which can fight pathogens, as well as antimicrobial peptides that restrict bacterial growth. Chemical action include enzymes that provide nonspecific defense. For instance, lysozyme, which is found in saliva and in tears and to some degree in sweat, destroys bacteria by weakening cell walls. When bacteria then can counter an osmotic shock, the cell wall is so weak that the bacteria rupture and die. Mucus traps pathogens and allows the pathogens then to be moved into the digestive system or out of the body. Gastric juice has a low pH and serves as a sterilant for the food you eat and the fluids that you drink. And it also contains an enzyme called pepsin, which is a powerful protease that works at low pH. So this is one of the hidden roles of the stomach, which is to sterilize stuff that you ingest. Interferon is a protein that causes uninfected cells to make antiviral proteins so that they don't become infected. These secretions, these interferon secretions, tend to work at a local level, so they tend to influence neighboring cells. If used in large amounts as a result of manufacture of interferon and ingestion or injection, it's effective as an antiviral, but it tends to tax the liver. So this is one of the reasons why interferons failed as a miracle drug to fight viruses and cancer, because it turns out that it's hepatotoxic. Phagocytosis is simply the act of engulfing and destroying microorganisms. Neutrophils and monocytes carry this out, as well as dendritic cells and macrophages and Kupffer cells. Monocytes entering the infected tissue become macrophages. They differentiate, and again, the purpose of this activity is not only to kill the pathogen, but also to present portions of the pathogen as antigens to warn our specific or adaptive immunity that we have a pathogen in the deeper tissues. Fixed macrophages remain fixed to a tissue. For instance, they'll be found in the bone marrow or in the spleen, while wandering macrophages wander among tissues and have a critical role in eliminating pathogens and warning lymphocytes that pathogens are present. So let's take a look now at the inflammatory response.
The inflammatory response is an important nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria.
Okay, fever. It's an elevation in body temperature as a response to infection. What does it do? It inhibits the growth of certain pathogens by making it too hot for them to reproduce, and it increases the rate of body processes, including those that fight infection. So basically, you cook the pathogen out. It's part also of the inflammatory response, as we already noted. Immunity is specific resistance directed at a particular pathogen or foreign cell. Production of specific cells and substances that attack a particular invader is accomplished by the lymphocytes. It also has a memory function. It allows then for a quicker neutralization of the pathogen in a second encounter than an initial encounter. And the reason for this is that as the attack against that pathogen is mounted, memory cells that are derivatives of T and B lymphocytes are stored in the body throughout life, and in the event that there's a second encounter with the same pathogen, they can mount a very rapid physical and chemical attack that keeps us disease free. So this is the purview both of lymphocytes and macrophages, again, always acting as a team. It's separated into two branches of immunity that we call cell-mediated and antibody-mediated immunity. Lymphocytes are generated in red bone marrow, which is found in the flat bones, in the ribs, in the coxal bones, in the vertebrae, and in the proximal heads of the femur and the humerus. They then specialize after they're generated from progenitor cells. In the thymus, they become T lymphocytes capable of coordinating cell-mediated and also humoral immunity. In bone marrow, spleen, or liver, they become B lymphocytes. So the B stands for bone marrow, the T in T lymphocyte stands for thymus. Okay, so that's how you can differentiate between the two broad classes of lymphocytes. T and B cells flow through the blood to lymphatic organs, where they generate large populations within those organs. 75% of circulating lymphocytes are T cells, 25% are B cells. And so what you're looking at here is sort of a diagram showing you where these cells come from and where they go. You can see here that stem cells in red bone marrow produce undifferentiated lymphocytes. Some of these will migrate through the circulatory system to the thymus, where they will be differentiated into T cells, either cytotoxic, regulatory, or helper Ts. Those are the three classes of T cells. The B lymphocytes, on the other hand, will mature in the bone marrow and then migrate to a lymphoid organ, where they'll mediate resistance by the production of antibodies. Okay, now we get to the process by which we identify harmful microbes in the body, harmful pathogens. First off, we should point out the fact that when we're talking about pathogens, we can refer to both living and non-living agents of disease. Examples of living pathogens would be parasites, bacteria, mold. Examples of non-living pathogens would be toxins and viruses, but they're all distinguished as a danger as a result of the molecules that are displayed on their surface, and we call those molecules antigens. So cells of each person has a unique set of identifiers on every cell in the body, and those are your self-antigens. Those identify the tissues and cells of the body as belonging to you, and so from the point of view of the immune system, those tissues are to be left alone, not targeted for attack. Lymphocytes learn how to distinguish your own antigens from antigens that are not found on tissues in the body, and this happens during the development of the immune system through a process of differentiation, DNA rearrangement, and clonal selection. And we'll get to those processes in a bit. This is what allows them to recognize foreign cells or infected cells in the body and target them for destruction. Each B and T cell has receptors that bind to a particular antigen. You have close to 1 billion different flavors of B and T cell in the body based on the antigens that they recognize, and that's sufficient to keep you disease-free for most of your life. Only those cells that can bind the particular foreign antigen are the ones that are going to be selected on the part of the immune response to multiply and then generate a standing army that will engage in a physical and chemical attack designed to wipe the pathogen out. The way that this happens, the way that you generate all these different specificities, is through a shuffling of segments of DNA during immune system development while you're still inside your mother's uterus that produces a variety of different combinations of nucleotides that can generate 1 billion different receptors on the cell surface, or 1 billion different antibodies secreted by lymphocytes designed to attach to and destroy anything bearing that particular antigen. Let's take a look at how the immune response actually operates.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Okay, so you can see here that there are two arms to specific immunity. And remember, this is anything that has gotten through our non-specific defenses. So our non-specific defenses are good, but they're not perfect, right? And those are our skin, our mucous membranes, their secretions, and the activity of granulocytes that generate a chemical attack against the pathogen regardless of its identity, and also the presence of the inflammatory response. In the event that pathogens get through those defenses, that's when we call on adaptive immunity to come in and try and save the day. So it's activated by the presence of foreign antigens either on the pathogen itself or on an infected cell. In cell-mediated immunity, what happens is that T lymphocytes recognize those presented antigens, and they are stimulated to engage in phagocytosis of the pathogen. Okay, the idea behind this again is that the T cells are going to secrete chemicals that will chemically destroy infected cells or destroy the pathogens, and at the same time attract macrophages to the area in order to engulf and digest cell debris and any pathogens that are in the area. This leads to a direct attack against the foreign antigens by chemical and physical means, and that would be the secretion of these chemicals that induce infected cells or pathogens to commit suicide, or that attract macrophages to the area so they can engulf and destroy the pathogen. We can destroy intracellular pathogens and foreign and diseased cells using this method. Remember that every nucleated cell in the body has the ability to engulf the pathogen and display bits of that foreign material on its surface as an indication that that cell is in fact infected. And that can be essentially like a signal flare used to call the T cells that are responsible for destroying the pathogen to the area so that they can engage in a chemical attack that destroys the infected cell. In the case of an actual pathogen, the simple presence of the antigen is sufficient to initiate a physical or chemical attack. In antibody-mediated immunity, it's a little bit different. What happens here is that B cells are activated as they encounter presented foreign antigen either on an infected cell or on the pathogen itself, and they're stimulated to then produce antibodies that are secreted into the bloodstream and are carried around the body by the beating action of the heart, and they bind to extracellular pathogens for destruction by phagocytic and by chemical means. Essentially, the antibodies are unique. If we were to sort of sketch an antibody here, let me draw an example of a typical antibody. We'll use blue as our color. Okay, and essentially what happens when the antibody is secreted is that it's going to have a particular antigen to which it binds very, very tightly. Okay? And so you can see in this sketch essentially how this works. So it's going to have the antibody is going to have oops, it's a little crazy there. The antibody is going to have sort of a Y shape to it. It's got a short chain and a long chain. And the binding sites are this area here and here. Okay, that's what attaches to the foreign antigen itself. So this whole thing is a protein. And when it attaches to the foreign antigen, it will render it inactive either by cloaking it, in which case it can't exert its toxic effect if it's something like an exotoxin, or it can engage in a process called agglutination, in which it joins together infected cells, causing them to form a large complex that comes out of solution and that can be easily phagocytized by wandering macrophages. Another method of attack is by essentially immunoprecipitating secreted toxins that come from the pathogen and rendering them inert and available for phagocytosis as well. They also serve to attract macrophages to the area through a system called complement. Complement proteins are always present in the plasma, and when antibody attaches to foreign antigen, complement proteins attach to the antibody, and this initiates a chemical cascade that ends up rupturing the cell membrane of the infected cell or the pathogen and causes it essentially to leak out and die. So those are just a few of the ways that these antibodies can work their magic. The nice thing about both of these arms of immunity is that in the process of generating cytotoxic T cells that can find infected cells and destroy them, and B lymphocytes that secrete these antibodies that can attack the pathogen at quite some distance from where they're produced, is that we generate memory cells at the same time, and these memory cells are essentially fully functional T and B lymphocytes that are selected again for their ability to bind that particular antigen, and they stay with you your entire life waiting for the second time that same pathogen shows up, at which point they'll very easily destroy the pathogen before it gets a chance to produce illness. So with cell-mediated immunity, the first step is to recognize the foreign presented antigen. Antigen-presenting cells engulf the pathogen and express part of the antigen on the plasma membrane along with a major histocompatibility protein, which you could think of as like an hors d'oeuvre tray, and the antigen being the hors d'oeuvre. The helper T cell can bind to the presented antigen, and then it secretes interleukins that will stimulate B and cytotoxic T cells that have the same specificity. The activated T cells divide rapidly for a T cell clone that's made up of helper Ts that recognize this particular antigen, cytotoxic and memory Ts, suppressor Ts, and also, we should note, B cells. Okay, so all of those different classes of lymphocyte are stimulated to multiply once we encounter the antigen presenting cell. Helper T cells start and intensify the immune response through the production of interleukins. They release these molecules when they're activated. That stimulates T and B cell clone formation. It also stimulates cytotoxic T cells to engage in their chemical attack on infected cells or actual pathogen. They can attack and destroy specific abnormal body cells by releasing a cytotoxin that ruptures the cell membrane of the target cell. We call this a membrane attack complex, or a MAC. They also generate cytokines that attract lymphocytes and macrophages to the area. And we already know that lymphocytes can engage in a direct physical or chemical attack on an infected cell or pathogen, and that macrophages are there essentially to engulf dying cells or pathogen if it's encountered. So that's essentially mobilizing your army in order to be most effective at the point of infection. The memory T cells are generated during the response, and they remain in the body for the entire lifetime of that individual, and in the event that same exact strain of pathogen is encountered again, it doesn't get a chance to produce illness because we have a standing army now of cytotoxic T and B cells and helper Ts ready to generate very rapid response to the second time the infection appears. So what you're looking at here is just sort of a diagram showing you how the T helper does its trick. You can see here that the cytotoxic T, or the helper T, binds to the presented antigen from the APC and then is stimulated to divide. And so we make many clones of a T cell that has this exact specificity. And then what happens? Well, these cytotoxic Ts can find infected cells, and the result then is they can generate a membrane attack complex that will cause the cell membrane to rupture and the cytoplasmic components to leak out and the cell to die. Okay, or at the same time, we can generate chemical signals that will attract macrophages. On the flip side, the T helper cell generates cytokines, and the cytokines are designed to stimulate rapid cell division of selected B and T cell clones in order to go out and fight this battle. So a B cell becomes activated when it binds to a specific antigen or is stimulated by the cytokines generated by the helper Ts. The activated B cell forms a clone of B cells that have the same specificity. The clone contains plasma cells that produce antibodies against the specific antigen, while the memory B cells again remain with the individual for decades in the event that same pathogen is encountered again. And so what you're looking at here is just a cartoon showing you the setup. So we have here antigen recognition by the receptors that are on the B cell membrane surface. This causes, again, stimulation of the B cell to divide. So we make many, many B cells with this exact same antibody producing capability. At the same time, helper T cells are also critical because the cytokines they produce further stimulate division, multiplication of these B cells, and then the B cells differentiate into plasma cells which create antibody in the plasma, and then that antibody can do its work either through agglutination or immunoprecipitation or through activation of complement. All are possible mechanisms whereby we generate a chemical attack that renders the pathogen inert. Antibodies form antigen-antibody complexes that mark the pathogen for destruction via complement fixation, which is the binding of complement proteins to the antigen. The complement, again, can rupture the cell membrane and cause it to burst, and the debris that is cleaned up by macrophages. Antibodies can also cloak bacterial toxins and block their actions on body cells, and the complexes can be engulfed by phagocytes, which render them inactive as well. The antibodies are called globulins. That's just sort of a general term for this class of proteins that has this particular shape, sort of a globular shape. The particular antibodies that generate immunity are called immunoglobulins, and they come in various classes as well. Some immunoglobulins are fixed to tissues via attachment to cell membranes. Some immunoglobulins are present in body fluids, and some immunoglobulins are primarily found in secretions. Okay, and they all have the same basic job, which is to attach to foreign antigens and tag it for destruction. The 6 basic ways that antibodies neutralize disease-causing ability from foreign antigens include the promotion of inflammation. Remember that inflammation is that process whereby the infected area becomes red, it swells up, it hurts, and the temperature goes up, and this is all designed to bring blood to the area. With the blood come the white blood cells that are going to fight the infection. Histamine is one of the major mediators of the inflammatory process. It also promotes phagocytosis. This is the engulfing of pathogens or infected cells and their digestion via lysosomal enzymes that renders them inert. And we can also activate complement, which attaches to antibody attached to foreign antigen and generates a membrane attack complex and also attracts macrophages. And those two processes, again, are designed either to destroy infected cells and tissues or to knock out the free-floating pathogen. There's also the mechanism of apoptosis. Essentially, antibodies can stimulate a chemical suicide trigger inside the infected cell or inside the pathogen that causes the cell to commit suicide, to slowly digest its genetic material, destroy its internal organelles, and shrivel away into nothingness. Agglutination, again, is the formation of large molecular weight complexes of antibody and infected cells that are then easily phagocytized, and it can also inactivate the pathogen or toxin by cloaking the portion of the antigen or toxin that produces the disease effect. So basically block it, interfere with its action. The primary immune response occurs with the first encounter of a pathogen. T and B cells make clones that destroy this antigen, and we produce memory cells that recognize the same antigen if it ever re-enters the body even more effectively than the first encounter. In the secondary immune response, we have a second invasion by the same pathogen that has the same antigen on its surface. The memory B and T cells that have already been generated are waiting for this second appearance of the pathogen, and they rapidly produce an immune response that destroys the pathogen in the infected cells before disease ever shows up. And so here you can see a graph showing you the primary and secondary response to foreign antigen. In the primary response, it takes close to 3 weeks to get the peak plasma antibody concentration from B cells that have been stimulated to produce those immunoglobulins, and then from there on, the amount of immunoglobulin begins to drop off because we're winning the battle against the pathogen. But in the secondary response, you can see that we get the peak antibody production in under 10 days, and it stays elevated for the next 3 to 4 weeks. The result is that there's never really any chance for the pathogen to generate the illness a second time. Now you might well ask, okay, why then do I end up with multiple cases of, say, a common cold or a flu throughout my life if my immune system is so great at defending against disease? And the answer to the question is essentially contained in the fact that when we're talking about pathogens, okay, we have to take note of the fact that pathogens also contain genetic material, okay, and that genetic material can be selected for in an environment designed to destroy them. Like, let's say we draw a little bacterial cell here, and let's say on the surface of this bacterial cell are some antigens. We'll just draw them like these little triangles. And with the initial encounter of this antigen with the antibody, we generate specific antibody via the B cells. We select for T cells that have the specificity, and we end up destroying this particular pathogen in the first encounter. Well, you can have a bacteria that has essentially a similar disease-producing capability, but generates different antigens on its surface. And what this does when it gets inside the body is it has to generate a new primary immune response all over again in order for it to be rendered inert, because now the surface molecules have changed. The surface molecules change because the genetic material that codes for them has changed as a result of natural selection. And so you end up with a second occurrence of the same disease, but it's not produced by the same strain of pathogen. Okay, so that's the explanation for that phenomenon. There's different types of immunity. The mechanisms are grouped into broad categories. In active immunity, the person generates the B and the T cells that provide the immune response. It keeps them disease-free, and it stays with them their entire life. It's much longer lasting than passive immunity, which is essentially the injection of antibody from another source that provides temporary resistance to a disease, but eventually that antibody, since it's not produced by immune cells in that individual, will be eliminated from the body. It'll be cleared, and then you'll be susceptible again. In naturally acquired active immunity, you encounter a pathogen, generate B and T cells to fight the infection, and then you recover, and the memory B and T cells are available then for later infection. In artificially acquired active immunity, you expose yourself to a vaccine. Now, what is a vaccine? It's not the pathogen itself, okay? These days, it's not even dead or weakened pathogens, as it says here in the bullet point. It's actually tiny molecules from the pathogen surface, antigens, that are derived from research into the types of antigens that are present on pathogens that are out in the environment at that point. And then we inject those antigens into the bloodstream, and you generate antibodies against those molecules that provides immunity against the actual pathogen should it possess that same antigen and infect your own tissues. So this triggers a response that generates memory cells that remain in your system for decades, and in the event that the actual pathogen shows up, it's as if it's a secondary immune response, and you're able to wipe that pathogen out. Booster shots can cause a secondary response to boost antibody levels even further, but again, you may have heard stories about people getting vaccinations and then getting sick after the vaccination, and those are true stories, but what's happening in those cases, in the majority of those cases, is that the person who gets sick after the vaccination is having a hypersensitivity reaction to the components in the vaccine, which produces symptoms very similar to the flu. Okay, um, the joints ache, you have secretions, you're tired, you may run a fever, and this goes on until the antigens are cleared, okay, rendered inert. And depending again on the individual, you could have an adverse event to any number of vaccines. In any large population, there's the possibility of an adverse event in a tiny percentage simply because the immune systems of every individual are different. While they're functionally the same in that they protect against disease, the the billion or so B and T cells in person A are not going to be the same billion or so B or T cells in person B that provide defense against disease. And there's the possibility among that billion or so that there happens to be a B or a T cell that will recognize and over-respond to that particular antigen in the vaccine and produce those adverse events. Some people never encounter a component in a vaccine that produces an adverse event, and so they have no problems. Naturally acquired passive immunity is where we have passage of antibodies from one individual to another. Examples include infants getting antibody through nursing on the mother's breast milk, or antibodies passing from the mother to the developing embryo and fetus via the placenta. And again, this lasts until the child's own immune system is capable now of actively fighting disease. So it's a prophylactic effect. And this is one of the reasons why children who are breastfed tend to have a healthier childhood than those that are brought up on formula, because formula doesn't contain any immunoglobulins. Now, in artificially acquired passive immunity, you get an injection of antibody from another source, could be an animal, could be another human being, and that provides immunity for a brief period of time until that antibody is turned over, is processed out of your system, and at that point you're vulnerable again. So for long-lasting, effective defense, the best method is either through vaccination or through naturally acquired active immunity. Now sometimes this can interfere with useful procedures designed again to keep you healthy, such as in tissue and organ transplants. The body can recognize the transplanted organ or tissue as foreign and attack it. And the problem then is that the transplanted organ or tissue will die, become necrotic, and become useless. So what we generally do before we do a transplant is we check the compatibility of the donor and the recipient using human leukocyte antigens, and what we look for is a 75% match between the donor and the recipient tissue. That's generally a baseline for the transplant taking, not being rejected by the immune system. However, we do have to engage in immunosuppressive therapy in order to sort of help the transplant along. Examples include cyclosporin, which inhibits T cell activity with minimal effects on B cells. The B cells are still able to generate antibody-mediated immunity against other pathogens as a result. Bacterial and viral infections are primary causes of death in individuals that are receiving immunosuppressive therapy. So this is a very tricky thing to do when you receive new tissue. To knock the immune system down, you always are going to leave yourself at risk for infection. So let's look at some disorders of the immune system. They can be grouped according to either infectious or non-infectious disorders. AIDS is an infection of the helper T cells by the human immunodeficiency virus, which is a retrovirus, which means it uses RNA as its genetic material. It attacks helper T cells, turns them into virus factories, and also causes them to express proteins on their surface that when they interact with other leukocytes and macrophages cause them to die. So your white cell count goes down very, very rapidly as the helper T cell gets hijacked into producing tons and tons of AIDS virus. So they invade these white cells as the virus particles are produced and ejected through the cell membrane. They attack other white cells in the area, and the immune defenses in that individual gradually waste away almost to nothing if the condition isn't treated. This leads to opportunistic infection, such as pneumocystis and Kaposi's sarcoma, which is a form of opportunistic cancer that the immune system normally knocks out in its job in immune surveillance, which is to search not only for pathogens but also for cancer cells. So, what do we do about this? There's no cure for AIDS at the moment, but there is a cocktail, and the cocktail consists of nucleotides that are incorporated by the enzyme that builds the genetic material for the virus, and this is AZT. You've probably heard of AZT if you've read magazines or newspaper reports of attempts to generate an effective treatment for AIDS. And then we also use protease inhibitors that are designed to interfere with the assembly of proteins into a virus capsid that would enclose the genetic material for the virus. And initially, these treatments were fairly effective, although they did put a heavy load on the liver. The problem now is that you're getting resistant strains of the AIDS virus cropping up, as you will with any drug-based cure for a particular disease. You're going to select for those strains of the pathogen that are not affected by that particular cocktail. And so this is one of the problems that we've run into with antibiotics, we run into with antivirals, is the phenomenon of pathogen resistance. Okay, infectious disorders occur by either a contact with infected body fluids. This can occur via sexual intercourse or through contact with infected blood, via sharing needles or exposed wounds or mucous membranes. And also, obviously, childbirth represents a method of transmission of pathogen from one individual to the other. Okay. Elephantiasis is another condition that affects the lymphatic system. This is a tropical disease found primarily in third world countries where public sanitation is at a minimum. Filarial worms are essentially, they enter the body through the ingestion of infected food or liquid, and what happens is these worms lodge in the lymphatic capillaries and vessels, and they impede the movement of fluid from the interstitium into the lymphatic capillaries, and the result is lymphedema. So what happens then is that the affected organ or limb swells up dramatically. This is transmitted by particular mosquito species that can harbor the filarial worm in their gut, also again by ingestion of infected material, food or water. The bottom line is that we do have a treatment for this. There's a drug called ivermectin, which is used as an ectoendoparasitic that can knock the worm out. The problem then is to resolve the swelling and also to resect the distended tissue. And in third world countries, that's often not an option. So frequently you'll see individuals with elephantiasis in third world countries simply because there's no availability of medical treatment. Tonsillitis is an inflammation of the tonsils, which are lymphatic tissue in the pharynx that contain macrophages and lymphocytes. Bacterial infection can cause the tissue to become necrotic, and that generally results in a tonsillectomy in order to remove the infected tissue so it doesn't serve as a source of disease. If the tonsils swell up and interfere with breathing, that warrants removal, or if it becomes a focal point for infection as a result of necrotic tissue, then we have to take the tonsils out. You have plenty of lymphatic tissue elsewhere in the body that can render you disease-free for most of your life. Okay, some examples of non-infectious disorders include allergies or hypersensitivities, which are basically an overly intense immune response to an antigen, which we call a trigger or an allergen. It's generally harmless to most individuals, but some people will have this essentially magnification of the inflammatory response when they encounter this trigger. The sensitization results in reactions whenever they're exposed to the trigger. The reactions can be immediate or delayed. An immediate reaction occurs when allergens bind with IgE antibodies on basophils and mast cells that cause them to release massive amounts of histamine that produces this inflammatory response that's widespread. Localized reactions are unpleasant, but they're not life-threatening. Examples include hay fever and hives. But sometimes we can get a systemic reaction, which we call anaphylactic shock, and this is life-threatening because it can occlude the upper airway and you can't breathe. It can also cause circulatory failure. Examples of triggers that can produce this, really anything is possible, but bee stings and penicillin have been reported. Again, this is going to depend on the individual and the composition of their particular immune system. Delayed allergic reactions appear 1 to 3 days after antigen exposure from cytokines that are produced by T cells. Examples include conditions such as poison ivy. Autoimmune disorders are a particularly nasty class of disorder, and this is a misdirected immune response against the body's own tissues. So T and B cells recognize body tissues as foreign, generate an immune response, and destroy that tissue. It can be due to a change in body antigens. It can also be due to exposure to a cross-reacting antigen, okay, such as an antigen on something that might not even necessarily be disease-causing, such as pollen, or maybe a mild infection with a bacteria or a virus. And we can generate then, in some cases, an antibody that recognizes not only the pathogen on the incoming entity, whatever it is, whether it's a pathogen or whether it's something relatively harmless, but can also cross-react if it's similar enough to your body's own antigens, and the result can be a misdirected immune attack. It's also possible that we can have a mutation within the immune system itself that produces an abnormal antibody or abnormal T cell receptor that recognizes now your body's own tissues where before it did not. These are all possibilities. Another possibility is that immune cells that are rendered inactive in a state of what's called anergy during the formation of the immune system, because they do recognize your body's own antigens, can suddenly wake up and attack your body's own tissues. So those are all possibilities, and all different types of causes have been noted and detailed in the literature. Lymphoma is any tumor of lymphatic tissue. This is basically white blood cell cancer. Hodgkin's disease is an example of a malignant lymphoma that causes enlarged lymph nodes, fatigue, and night sweats. Chemotherapy and radiation yield a relatively high cure rate. There's also a host of drugs that are used to treat these white blood cell cancers. Now, the thing about these white blood cell cancers is that the individual that has them is going to have a very high white count. However, they're going to be immunocompromised, and the reason for this is that the white cells that are produced via the cancer are not immunocompetent. They don't have the ability to fight disease, and so they essentially clog the system for the immunocompetent lymphocytes and macrophages, and the result is increased incidence of infection and opportunistic cancer. Okay, that brings us to the end of this podcast. Again, make sure and review this material, as it will be covered on the exam and in your homework. And I'll see everybody in the next podcast. Thanks for listening.